All right, hi everyone. I'm Leyna Stemle. I'm a PhD candidate at the University of Miami, and today I'm going to be talking to you about our collaborative work with Archbold entitled Explaining Seasonal and Habitat-Specific Growth of Immature Gopher Tortoises. Is this a job for drones? All right, so gopher tortoises live in a variety of different habitats. Some are natural, like flatwoods, sand, hill, and scrubby areas, and some are disturbed habitats, like you see here, open grassy rural areas, roadsides, things like old agricultural and pasture lands as well. But we know that these habitat differences impact growth of these tortoises. We even have one example here at Archbold from long-term data sets looking at adults, where the adults in the ruderal habitat in Hill Garden were larger than those in the sandhill. So you can see that here from my lab mate Hunter, where adult females had a larger carapace length or shell length in Hill Garden, the rural habitat, compared to sandhill, as well as higher annual apparent survivorship. But we don't have as much information on young gopher tortoises and how their growth is impacted by different habitat types. But these young age classes are really important to be understanding gopher tortoises as a whole and how to manage them as well in the land that they're on. One example of why gopher tortoises that are young are really important to the demographics of gopher tortoises is size at sexual maturity. If they grow faster and reach size at sexual maturity at a younger age, they then have more years where they're able to be reproductively active and increase their lifetime fitness. So at Archbold, we had done movement studies of young gopher tortoises, and in the field we were noticing visual differences of these young tortoises and the different habitats. So here's a tortoise in Hill Garden, which is a grassy area, compared to one in the sandhill, and they're the same age, but you can see the sandhill one is much larger. So we noticed these habitat-specific size differences, and we're wanting to see how that was impacting growth and how we could look at management questions for these tortoises, like how does young tortoise growth vary in relation to food resources? So of course we did our study here at Archbold, and my lab mate Hunter had analyzed some previous data on adults, but we wanted to look at the young ones. So for the sake of this talk, Hill Garden is the rural grassy area, and all of the surrounding units were considering sandhill. So to track the tortoises and their growth, we had to capture them quarterly. To make this easier, in early 2021, we captured 27 of them and radio tagged them. This way we could follow their movement so we could know if they're changing habitats, so we know what food resources are actually around them as they might move different burrows. We captured them quarterly to get the morphometric data, the growth data, until the end of September 2022, so that ended up being 6 different capture periods. We gathered a lot of morphometric data, but the main one we're focusing on is carapace length or shell length and relating that to growth. In 2021, we did on-the-ground vegetation surveys, but for all of the seasons, we had red, green, blue drone data, and then for the last 2 seasons, we had multispectral drone data. So our objectives were to test if these young tortoises were having different growth rates between the 2 habitat types, and also see if we could find what predictors were the best for growth. We also wanted to see how useful the drone data was in relating to growth and for studies like this, as well as how these young tortoises were doing in non-native habitats. So again, for all of the seasons, we had the red, green, blue data from the drone imagery from the GIS lab. And from this, Julie was able to do supervised classification into these 5 categories here. So the top is the ruderal habitat and the bottom is the sandhill. And these circles are 14 meters around each burrow that a tortoise used, which we're considering the feeding radius for the tortoise. You can see for ruderal habitat that the green herb category is the most dominant, and that includes things like mostly non-native grasses. Whereas the sandhill is dominated by the dark green, which is woody vegetation, the purple, which are trees, and the tan, which is bare ground or sandy open areas. We then did similarly on-the-ground plant survey data to compare to this and to compare to growth in 2021. So for 3 of the seasons, again using this 14-meter buffer and the 42 1-meter square plots this time instead of the whole buffer from the GIS data. So the 1-meter square plots, we determined presence and absence of these vegetation and food resource categories, including wiregrass, bahia grass, exotic legumes, native legumes, and different categories of woody vegetation. As I mentioned, we had red, green, blue drone imagery from all of the seasons, so we were able to do spectral analysis, including some vegetation indices, because these are often related to nitrogen content of the plants, which is then related to tortoise growth. So we did these two for every season, and then in the last 2 seasons, we had the red edge drone data, so we were able to do a lot more multispectral indices, including things that you may have heard of, like the Normalized Difference Vegetation Index, or NDVI. Now moving on to some results, here's a repeated measures ANOVA looking at 2021, so it's broken up by season as well as by habitat. So hill garden is red and the sandhill is blue, and on the Y is a metric of growth, so shell length growth % per day. And you can see that hill garden in every season, the tortoises had higher growth than in the sandhill, and there's this kind of downward trend across the year as there is a season effect as well as habitat effect. So most of their growth is occurring between March and September, with some happening after into December. Now for 2022, we see the same pattern where there's a habitat effect. So hill garden had higher growth than the sandhill in both seasons as well. So here are the 2021 broken up by season, the data. So I analyzed it 3 different ways. One was the drone supervised classification data that I mentioned from the red, green, blue imagery. This one is the on the ground plant survey data. And then this is the drone spectral indices. And there's the top model for each of these is in the box. And the top model comparatively per season is in dark green. So like for these 2 seasons, the best model, so the lowest AICC, was plant survey data, but for late wet season in 2021, the drone supervised classification had the best model. Overall, all of the models are saying a similar thing, that non-native grasses like bahia grass are positively related to young tortoise growth. So here's a broken down for the late dry season from each of the top models. You can see that growth is going to be on the Y and the habitat variable is on the X. And the supervised classification green herb category is positively related to young tortoise growth with a pretty strong relationship. And similarly, from the on-the-ground plant survey data, our bahia grass presence and absence variable has a strong relationship to tortoise growth as well. It's just a little bit stronger. Now looking for the spectral indices, the green leaf index was the one that had the top model in the late dry season of 2021. This is the negative values are the brown values, so the smaller ones are like grass, whereas the higher values have more chlorophyll, more green leaf index, so things like small trees and dark colored green that are more in the sandhill. And so it makes sense that we see the opposite relationship where young tortoise growth is negatively related to the green leaf index, because a higher green leaf index is more related to woody vegetation presence, which is not very good forage. Now here's the same kind of table for the 2022 season, but instead of on-the-ground plant survey data, we have this other category, multispectral drone indices, because we had that new layer added. Here we see the drone supervised classification did really well again, and then the multispectral indices did better than the spectral indices in general. All of these data are showing us the same trends, that grasses, including non-native vegetation and grasses, are positively related to young tortoise growth, whereas dense woody vegetation, like you might see here in like 5A, where there's a ton of young oaks that are quite dense, that's less positively related to growth, so it's negatively related to growth. For these different methods, they had a different time investment, but they were all kind of telling us the same thing. Again, we had to have someone that could fly a drone, we had to have a drone, and we had to have computers that could handle the data and handle the processing and the runtime of all of this really high resolution drone imagery. So supervised classification and spectral indices, the person hours weren't a ton, but the computer runtime was quite long. But compared to plant survey data, on the ground, we had to teach people how to identify all of the plants, do all of the 42 1-meter square plots for every burrow that a tortoise went to in that season, and then every season had to do them again, and then entering that data by hand. So that took a lot of person hours, whereas the drone data took a lot of computer runtime and things like storage and things. But we were really happy with the results that we got from comparing the plant survey data to the drone data. So on the X is the drone supervised classification category green herb, that's indicative of non-native grasses. And on the Y, we have Bahia grass presence from our time-intensive on-the-ground plant surveys. And they have an R-squared of 0.95, so that was pretty impressive, and we were really happy with that. Quickly, just some movement insight that we got from this study, because we were tracking the tortoises also. For a year, 3 of the tortoises were staying up on Red Hill in the restored sand hill, but then in the late dry season of 2022, in May, they all started to go downhill towards the station to unrestored sand hill, moving very far different distances, and one actually moved back a little bit. But we think this might be due to availability of food resources and weather, because in March, April, and May of 2021, there was more rain than in March, April, and May of 2022, when they decided to then head back down. But these long distance movements haven't been recorded for this age class, so it was really cool to get kind of high resolution data for movement of these tortoises as well. So overall, the non-native plant communities that are on Hill Garden are positively related to young tortoise growth. But the small-scale habitat heterogeneity on Red Hill was contributing to this within population difference and age-specific body size and possibly age at maturity that we were noticing in the field. But we're still trying to understand the potential trade-offs of growth in rural habitats. There could be things like bone density or survivorship that may be impacting the tortoises that we haven't been able to tease out yet. But overall, the drone data was really promising, and it was really useful for us to kind of identify the forage of young tortoises in a very high resolution way. And it could be helpful to manage recipient sites of tortoises that are being translocated, or remnant populations of tortoises that are quite big areas, and you just want to get an idea of what forage is available for the young tortoises. So with that, we have a lot of people to thank, as it takes a lot to catch 30 young tortoises 6 or 7 times and get all of their measurements, as well as all of the drone data. So of course, I'd like to thank our funding, Disney Conservation Fund, and the University of Miami Cushman Grant. So with that, I'll take any questions if we have time. It looks like there's one on the Zoom. Yes, so they didn't all survive.
Leyna, before you start, I'm going to take one from the floor, and then we'll read out the question on the Zoom so everyone can hear it. So Sahas, would you like to go first? Thank you.
Hi, Leyna, thanks for a wonderful talk. So did you control for differences in densities of tortoises at the different habitats to look at growth rates?
No, we did not control for density of tortoises. This is just kind of an initial look at one of my dissertation chapters, but that is something to consider as well.
Thank you.
Okay, Laura, would you like to read the question from the chat?
Yep, Fitz says, fantastic study. Wait a minute. Did you learn anything about survival of these early age classes, or did they all survive?
Thanks, John. Yeah, we actually did, and I'm still analyzing some of that data initially, but they didn't actually all survive in our study. We had one that died and a few that disappeared. You know, radio tracking, we couldn't confirm what happened to them, if they dispersed or if they died. But we did get some good survivorship data for these age classes, which is going to be important to the literature, I believe, as there's not a ton of that data available. But it was kind of higher than we were maybe thinking. But we were tracking tortoises that were mostly above 3 years old. So a lot of the ones that die are between, you know, just hatching and like 1 or 2. So we weren't tracking the really small ones for this study.
Okay, any other quick questions? No, if not, thank you very much, Leyna, and thank you for joining us. We really appreciate it.
Yeah, thanks for having me.
To go on to our next speaker, Laura, are you sort of vaguely set up here?
She's on it.
Oh, we're already on. Well, why don't, in the interest of time, why don't we just, we don't see her face yet.
She just has to talk.
Okay, just if you start talking, Jenny,
Sure. Sure. All right. Thank you.
Okay, so Jenny, there you are, and we can all see you. So
Okay.
over to you. Please introduce yourself and introduce your talk. Go ahead. Thank you.
All right. Thank you. So I'm Jenny Schafer. I'm an assistant professor at Winthrop University in South Carolina. Today, I'm going to talk about research I conducted with an undergraduate student investigating an indirect defense mechanism in Chapmannia, floridana. This research does not directly relate to any of the 4 pillars that Hillary mentioned this morning, but I hope you'll find it to be at least a somewhat interesting natural history story. All right, so plants have a limited amount of resources that they can allocate to growth, survival, reproduction, or defense. By defending photosynthetic tissues, plants can increase photosynthesis and acquire more resources. So plants have several types of direct defenses that they can use against herbivores, such as a thick waxy cuticle on leaves, or trichomes, which are hairs on the leaf. And these can deter more commonly insect herbivores. Plants can also have chemical defenses, such as secondary metabolites, including things like caffeine, nicotine, or phenolics. Plants can have modifications of structures. So in cacti, leaves are modified into spines to defend against herbivores. And some species can have branches or stems that are modified into thorns. And so all of these are examples of direct defenses. Plants can also have indirect defenses. Probably most famous example of this is acacia trees in Africa. They have swollen areas at the base of the thorns that are called domatia that provide a house for ants. They also provision the ants with food sources. And so ants are attracted then to live inside the acacia plants. And when herbivores come along, the ants will run out and bite their faces and deter herbivores from eating the leaf tissue. So this is an example of an indirect defense. Another more recently recognized example of an indirect defense is glandular trichomes. So these are sticky hairs on leaf surfaces. These sticky hairs can catch or trap small insects that are flying by the plant, and these insects then die on the plant, and so they would be classified as carrion. And the carrion can attract predators such as spiders or assassin bugs to the plant. They would be scavenging for these food resources. And the presence of predators then would have a negative impact on herbivores by eating them directly, and so limiting their abundance on plants or deterring them from feeding on plant tissues. And then herbivores, since they have a direct negative impact on reproduction by consuming reproductive structures such as flowers or fruits, predators then have an indirect positive effect on reproduction, such that glandular trichomes then can have an indirect positive effect on plant reproduction. And so we investigated if glandular trichomes are functioning as an indirect defense in Chapmannia floridana. Specifically, we tested the predictions that the number of trapped carrion is positively correlated with the trichome covered stem length. You might have noticed when you're out in this in the field at Archbold that the reproductive stems have these glandular trichomes all across the stem and fruits. We also tested the prediction that the probability of damage to reproductive structures is negatively correlated with the number of carrion that are trapped on the reproductive stems. So to test these predictions, we haphazardly selected 84 stems in 6 vegetation types. So Chapmannia floridana occurs throughout Archbold property, including Southern Ridge Sandhill, Florida rosemary scrub, and scrubby flatwoods, but also in degraded scrub, fire lanes, and pastures. And so we sampled individuals across all of these vegetation types or habitats. And we measured the height of the reproductive stem, the length of the trichome covered portion of the stem. So the trichomes typically start at some point on the reproductive stem. And so we considered what portion of the stem is covered by trichomes. And for a subset of individuals, we counted the number of trichomes on 3 stem sections near the top of the plant, closer to the majority of the reproductive structures, in the middle of the stem, and at the bottom of the stem. We also then counted the predators and herbivores on the stem and the carrion that were trapped on the glandular trichomes. And then we checked for damage to flower buds, flowers, and fruits. So in this figure, the vegetation type is on the x-axis with the sandhill, rosemary scrub, scrubby flatwoods, degraded scrub, firelands, and pastures. And the total reproductive stem height is on the y-axis. We found that there is a variation in stem height across vegetation types, with shorter stems found in pastures, which could be related to disturbance type and frequency with having both fires and herbivory by cattle. But despite these differences in reproductive stem height, we found that there was no difference in the trichome covered stem length across vegetation types. So this suggests that reproductive stems across vegetation types have kind of a similar capacity to entrap carrion based on the portion of the stem that is covered by trichomes. This figure is showing the number of trichomes on the x-axis and the location on the stem on the y-axis. And we found that the number of trichomes increased from the bottom to the top of the stem, and this pattern was consistent across vegetation types. And so there's a higher concentration or abundance of the glandular trichomes near the top of the stem, where many of these reproductive structures are located. In regards to carrion, we found trapped carrion on 74% of the reproductive stems. The most common groups that these carrion came from were the insect orders Coleoptera, Diptera, and Hymenoptera. This would be beetles, flies, and ants. And this picture is from the year before we did the study, which is what got me interested in this research in the first place. When we did the study in 2019, we didn't find stems that had quite this many trapped carrion, but this is just to show you an example of kind of the potential of these stems to trap these small insects that are moving through the environment. And so in this graph, again, vegetation type is on the x-axis and the number of carrion trapped is on the y-axis. And this is per centimeter of the trichome covered portion of the stem to account for potential variation in the number of trichomes on stems. And we found there's no difference in the number of trapped carrion across vegetation types, which suggests that the abundance of these small insects moving around these habitats is similar, such that there is a kind of similar potential to trap carrion across all these habitats. And here, the x-axis is the trichome covered stem length. The y-axis is the number of trapped carrion, and there's a positive correlation between these two variables. So as there's a kind of a longer length of the stem with glandular trichomes, more carrion are trapped on those stems. In terms of predators, we found predators on 6% of reproductive stems. All the predators we found were spiders, most commonly green lynx spiders, which is in the picture on the left, and crab spiders. We found herbivores on 37% of reproductive stems, and most commonly we found grasshoppers, caterpillars, and hemipterans, such as leafhoppers. Overall, a third of the reproductive stems we sampled had some type of damage. Of the plants that had flower buds, 21% of those buds were damaged. Of the stems that had flowers, about 14% of those were damaged. And of the plants that had fruit, about 11% of those were damaged. This figure is showing the number of trapped carrion on the x-axis and the probability of damage on the y-axis, where 1 indicates damage was present, 0 indicates no damage. And the size of the circles is scaled to the number of stems that have that specific number of trapped carrion. We found that there was a negative correlation between the number of trapped carrion and the probability of damage. So as a plant is trapping more carrion, providing more potential food sources For predators, that's associated with a decline in the probability of damage to reproductive structures. So we could conclude that glandular trichomes appear to be functioning as an indirect defense in Chapmannia floridana. Our results don't necessarily exclude other potential functions. So the glandular trichomes are also likely providing a direct defense by trapping smaller insects such as aphids and spider mites. But there does appear to be an indirect defense function of these glandular trichomes. We were able to show that as the abundance of glandular trichomes increased, there is an increase in the number of carrion that are trapped. And on stems that have more carrion, there's a decreased probability of damage to the reproductive structures. Further observations and experimental studies that focus on these other links in regards to predators and herbivores could provide some more support for our conclusion. Other studies have looked at the relationship between the number of trapped carrion and the number of predators on plants, but these studies have often been conducted in plant species that are more shrubby and have glandular trichomes on the leaves and non-reproductive stems. And so we only typically found 1 or 2 spiders per stem, so we were not able to look at that relationship. Could also investigate the impact of predators on herbivore abundance. So we just looked at presence, absence, and the direct effect of herbivores on herbivore number on amount of damage. So one final thing I want to note is that we did in one case observe a lynx spider that had caught a pollinator. So there can be some potential cost associated with glandular trichomes trapping carrion and attracting predators. Trapping pollinators could reduce pollination, but the fact that Chapmannia persist with these glandular trichomes suggests that the potential cost of reduced pollination are lower than the potential benefits of trapping carrion that provides defense, attracts predators that provide defense against herbivores that are targeting these reproductive structures. And so I'd like to thank the Archbold Plant Ecology Research Program. This research was conducted when Eric Menges was running the lab. Dr. Paula Mitchell helped with the arthropod parts of the research, and this research was funded by a Winthrop University Research Council grant. Thanks. And I'm happy to take any questions if I have a second or two.
Well, great job, Jenny. Thank you very much. Before we open the questions, I'm going to add a couple of points. You need never apologize if your research does not fit into one of those pillars. Of course, we welcome all research and it builds on knowledge. So there's never an apology needed for that. And the second, I did want to point out to you, I remember Tom Eisner, Mark might remember this, years ago, we were having a small party for him in the late 1990s. He said, on reflection, many of the things I've worked on at Archbold have actually been stitched. So you're following a very long tradition of that. Okay, we've got a question from the floor, Reed Bowen, and then I'll follow up with some others.
Hey, Jenny, great talk. Is there any evidence that plants that use glandular trichomes actually benefit from the carrion? Do they absorb any of the nutrients from the carrion?
Yeah, a study was actually just published, I think in 2021, and this was recognized as kind of a new idea for plants like this, particularly ones, and so this was found in a species in Washington. It's kind of a similar structure to Chapmannia, so this is another thing that would be cool to investigate. It has just the glandular trichomes on the reproductive stem and has like this one long stem like Chapmannia does, and so they did find that the plant was absorbing nitrogen from the carrion through the glandular trichomes. Yeah.
Next question, Sterling.
Is there any specific chemical in the glands that attracts these insects, or is it just kind of random?
I'm not sure that there's any specific chemical that attracts the carrion. I haven't read anything about that. But there are potentially, there could be chemicals in the glandular trichomes that directly deter herbivores that might be if they move across those and release the chemicals or eat something that has them. A lot of these glandular trichomes do produce or house secondary metabolites themselves. So those are both great questions, hitting on the other potential functions of the glandular trichome. So great questions.
Yeah, go ahead, Matt.
My question also has to do with the other possible functions. I know that, trichomes might be a pathway for foliar water absorption. So given that it's an xeric environment, do you think that these trichomes have any function as a water regulatory mechanism or a drought resistant mechanism?
That I am not sure of. I haven't come across any studies that have specifically looked at that I'm aware of, but that's a great idea as well. They are kind of moist. They could maybe trap some moisture along the stem as well. So I'm not sure about that.
So one last question, and you might shrink while you're answering this as we move on to the next speaker. So go ahead, Aaron.
Yeah, I thought it was a really cool talk. I was curious that relationship between the carrion and the herbivory, if there's other alternative explanations that it could be, and if you have or thought about doing like an experimental removal or addition of the carrion.
Yeah, so I mean, we specifically were trying to look at if carrion were potentially attracting predators and reducing herbivory. There are, I guess, other explanations for that relationship. And the next step, yes, would be experimental studies. So that would be something that could be done. And a lot of other studies, research has done those experimental studies to more clearly show that link. And I don't know how to change my size or if you said to shrink myself.
Don't worry, we've done it for you.
Yeah, I was doing it here. You do have 2 online questions. If you'd like to go into the Q&A
Sure.
and just type the answers, that'd be great.
I can do that.
Thank you so much, Jenny. We really appreciate it, and nice to see you here,
Yep.
at least virtually. We're going to go on to our next speaker, who's here in person. So I'll let you introduce yourself and go ahead and get your photo. Thank you. Thanks.
Okay, good morning, everybody. My name is Andee Naccarato. I'm a relatively new research assistant in the plant ecology program here at Archbold Biological Station. And I'm honored to be able to share some recent results from a long-term study and experimental introduction of Crotalaria avonensis, a rare endemic plant of the Florida scrub. Crotalaria avonensis is commonly known as Avon Park harebells. It is a short-statured perennial legume that grows on xeric white sands on the Lake Wales Ridge. It can reproduce clonally and through seeds. However, this is one of the most imperiled plants on the Lake Wales Ridge. It's endemic to the Lake Wales Ridge, and it does have federal and state protections at the endangered level. So you may be thinking, this is such a cute plant, how could it become so imperiled? That's what I thought. But Avon Park harebells has 3 different categories of conservation challenges. That first category has to do with its biology and ecology. It is a reproductively challenged plant. Only about 10% of its flowers result in seed pods. After that, chances are very low for seedling recruitment. And if the seedling does happen to grow to an adult plant, it seems to be at the top of the menu for mammalian herbivores to eat the entire above ground portion of the plant. But thankfully, Avon Park harebells can resprout after herbivory and after fire events, which do impact its habitats. The second category has to do with its geography. Like I mentioned, it's endemic to the Lake Wales Ridge. But even more limited than that, Avon Park harebells is only found in a portion of the ridge, northern Highlands County and southern Polk County. And then beyond that, these plants are only found in 2 protected preserves and 1 unprotected subdivision. Then that leads us into its last category of conservation challenge that has to do with human activities. Of course, there has been degradation and loss of upland habitat across the Lake Wales Ridge. You're seeing an example of a small portion of that here. This is an aerial view of that unprotected subdivision where Avon Park harebells occurs. And since this population started to be monitored, the proportion of plots that still have plants has steadily gone down with time. Only about 20% of plots still have plants as of last year. So you can see this unprotected population is under extreme threat of extirpation. Now we're looking at an aerial view of one of the protected preserves. You can see this piece of land was not always a preserve. It was platted for development at some point. And there are private end holdings in this area too, including one area that's directly adjacent to one of our sites that has Avon Park harebells, and it is slated for development in the near future. So members of the plant ecology lab went out and put up extra fencing and extra flagging to try to show there's something very important right here. Please don't run this over when the development starts to happen. So you can see that even in the protected areas, they're not safe from threats. So what can we do to stop the Avon Park harebells from going extinct? What you're seeing here is a conservation strategy map, and I'm not going to go through all of the boxes, but it is important to know that after decades of monitoring the 3 wild populations, it's become very apparent that new populations are needed through translocation plantings to secure the species' persistence. So back in 2012 and 2015, 2 introduction events were planned for Avon Park harebells at the Silver Lake, Lake Wales Ridge Wildlife and Environmental Area in Avon Park. That's managed by Florida Fish and Wildlife Conservation Commission. The introduction locations were based on appropriate soil type, habitat, and microhabitat. And the management units for the introductions were burned a few years before those introductions took place. So as the introductions were being prepared, there were a couple of major research questions on the mind of the plant ecology program here. Number 1 is survival influenced by introduction technique, namely a seed introduction or a transplant introduction. And second, what roles do biotic and abiotic factors have in the introduction success? And these are factors such as habitat type, microhabitat factors, as well as seed treatment before the introduction. So let's take a closer look at the Silver Lake tract where these 2 introductions took place and zoom in on the 2012 introduction site. This one was both a seed introduction and a transplant introduction. The red circles that you see are the start and end of 5 different transects, and a total of 400 seeds were introduced along those transects. They were either in the more open rosemary scrub habitat or the denser scrubby flatwoods, and half of the seeds were scarified prior to introduction and half were not. And then the yellow triangles are the transplant introductions. There were 84 transplants of 3 different propagation types. There were wild plant cuttings that were propagated by Cheryl Peterson at Bach Tower Gardens, and tissue culture cuttings and tissue culture grown plants from Valerie Pence of Cincinnati Zoo and Botanical Garden. And if we move on over to the 2015 introduction site, this one was a bit simpler. It's a single transect of, again, 400 seeds that were introduced. There were 2 experimental variables of interest here, whether the seeds were sown on top of the sand or planted at a 1 centimeter depth, and then whether or not native leaf litter was introduced into the plots. These is just a couple of pictures of the 2 different introduction types. On the left is a typical seed introduction site. A single seed was placed in the middle of that PVC pipe that was set into the sand. And then the whole thing was encircled in herbivore exclusion caging. And then on the right, that's a transplant introduction. No PVC pipes needed here. And the herbivore exclusion cage was taller and open on the top to allow for the plant growth. So ever since the first year of each of those introductions, members of the plant ecology program have gone out at least 3 times a year, if not more, to monitor all of the plots for plant survival, growth, demographic status, and recruitment. And towards the end of 2022, we went out to do microhabitat assessments for the 400 seeds and the transplants in the 2012 study. And the microhabitat assessment looked at things such as percent vegetation cover over each plot, as well as plant species composition surrounding the plots. So let's get into the results. The first question had to do with whether a seed or transplant introduction was going to be more successful. And just at first glance here, at the left is the seed introduction. You see that going down pretty steadily over the years. As of last year, only 12.1% of the seed germinants were still alive. And although there has proportionally been increases in the number of plants flowering with time, which is encouraging, no second generation seedlings have been found after 10 years post-introduction for the seeds. But on the right, for the transplant introduction, looking more positive over here, 33% of the original transplants were still alive as of last year. There's been an increase in the number of individuals, mostly through the clonal recruitment, but perhaps most excitingly, 24 seedlings of the second generation have been found around the transplant sites. The transplant type itself, though, did not have any effect on the survival of the transplants. Now let's take a look at just the first seed introduction. They were sown into 2 different habitats, and the seeds were either scarified or not. And we can see here that habitat did have a significant effect on germination success. It was increased in the scrubby flatwoods habitat, so that's the slightly denser habitat, and seed scarification did not have an impact on either total germination or survival of germinants after the 10 years. Now let's look at the microhabitat, the percent vegetation cover variable. So we got a couple of graphs here. On the left, it's percent vegetation cover and its impact on germination success. And then on the right is the impact on cumulative survival of those seeds that did germinate. So you can see on the left here, the plants that did germinate were associated with approximately 20% vegetation cover, although there is a range from 0 to 60% vegetation cover. And then if you look on the right, about 10% vegetation cover is what's associated with the currently surviving seedlings. Although the sample size is lower, only 23 of the germinants were surviving through last year. And there is a similar range in values of percent vegetation cover between those surviving and those that did not. Finally, the second seed introduction was looking at the effects of seed planting depth and presence or absence of leaf litter in the plots. And we can see here that both of those variables had an impact. Germination was improved when seeds were buried to 1 centimeter in the sand, or leaf litter was present in the plots. Interestingly, that interaction didn't have a significant effect. And unfortunately, only 6% of the germinants survived until last year for the second seed introduction. So let's focus on the positives and the accomplishments. The Plant Ecology Laboratory and its partners have increased the number of population sites for Avon Park harebells from 3 to 4. And we learned that using transplants is the more successful introduction technique, and it's currently recruiting its second generation, which is very exciting. Then we learned some more specifics about conditions that can improve germination for this very rare plant. Seems to prefer the scrubby flatwoods habitat with approximately 20% low vegetation cover, and the seed should be buried or leaf litter present. As for future directions, the plant ecology program will continue monitoring all of these populations and pay specific attention to that protected population that does have adjacent development happening in the near future to monitor the response that it has to that disturbance. And there are plans just getting started for another transplant introduction to hopefully result in even more population sites. So I hope you've learned a little bit about Avon Park harebells and its current conservation status and directions. And since this was a long-term study, there's a lot of important people and organizations to acknowledge for its success to this point. And I'll be happy to take questions, and thank you all for listening.
So we've got a minute or so for questions. Are there any from the floor? Go ahead, Meredith.
So what prevents it from growing on the station?
That's a really good question of what prevents Avon Park harebells from growing on the station. I've thought about that myself, and as of now, I don't have an answer. Does anyone else in this room have an answer?
Are we planning to do any translocations here?
I don't believe Archbold Biological Station has been considered for a translocation site. The translocations are wanting to stay within the narrow known native range of the species.
Yeah, so we're going to swap over the talks, but maybe there's time for Andee to ask one more question. If anyone has one, Betsy.
Do you have to consider any of the genetic diversity of the plants when you do a translocation?
That's a very good question. For the introductions that I was just talking about, I know the source material was collected from the unprotected population. But beyond that, I don't have an answer for you, but I can try to follow up.
Okay, well, thank you. That's great talk. Okay. So our next speaker, Greg, go ahead, introduce yourself and your talk. Thank you.
Hi, everyone. My name is Gregory Sonnier. I'm an assistant research biologist at the ranch, working with Betsy. For those of you that have been around for quite some time, you know that focus is heavily on wetland and wetland biodiversity, how management of ranchland affect wetlands, and also working on wetland restoration easement outcome. And I'm very sorry to go from like, let's do everything we can to save this species to let's do everything we can to kill that one. So I'm sorry about that. The title of my presentation is Cattle Grazing as Alternative Management of West Indian Marsh Grass, Latin name Immature amplixicalis, in subtropical wetlands. Here, I'm sure you're fully aware of the problems related to non-native invasive species in Florida. We kind of have a hotspot for those species. And here are just a couple, like two from the animal realm and two from the plant realm that are really well known: Burmese spider, water hyacinth, Kogan grass, zebra mussel. And it's a worldwide problem as well as a problem in Florida. The non-native species tend to affect native ecosystem by outcompeting native species. They can create some other issues related to ecosystem functioning. And they have evidence that they could have potential negative effect on fire intensity and fire return, for example, for Kogan grass. And I'm not going to talk about any of those four species. I'm going to talk about a species you might not have heard about, which is Chapmannia amplixicalis, which is right there. Why I'm talking about this species, it's a relatively recent non-native invasive in Florida, in central Florida and southern Florida, and it really invaded heavily ranchlands. For the record, at Archbold Biological Station, Bacalon Ranch, our first record, the earliest one, is from 2006, when he started the long-term USDA wetland experiment that Betsy started in 2006. Why is it a species of concern? It's classified as a Category 1 invasive by the Florida Invasive Species Council. It's an obligate wetland species. It is capable of forming monospecific stand. It will outcompete native species very quickly. And it's an efficient disperser. So we had some work that was done by Elizabeth Bouchard, or couple, like in 2018, that was published, a research intern that looked at what were the landscape factors that were affecting this dispersal of that species. And she found evidence that the ditch network was increasing the likelihood of finding that species in seasonal wetland. The literature says also, and we'll get back to that maybe at the end if I have time, that it's capable of forming a persistent seed bank. So keep that in mind. If I have time, I can talk about that at the end. And as I mentioned, it's a heavy clonal growth, a high vegetative spread. Just to give you an idea, this is me, I'm not the smallest guy in the room, and you can just barely see me in the middle of that wetland that's kind of invaded by West Indian marsh grass. And the problem is that when you spend a lot of money on wetland restoration easement and you have a species such as West Indian marsh grass coming in, you might end up with really preventing the success of your restoration. So it becomes very important to control that species. What are the different methods of control? Like the first method is like physical method, like removing the species from the ground, but that only works when it's early stage of invasion, when you have only a few individuals. It's not really feasible at larger scale. So the other alternative is to use chemical application, and like the two big ones are glyphosate and imazapyr. The problem with chemicals, that's there's a significant cost associated with it just for the labor and the cost of the herbicide, and there might be some concern for environmental integrity. There's another alternative, which is biological, which is one aspect of West Indian marsh grass that's kind of cool, is that it's highly palatable, highly digestible, and it has high nutrient and high crude protein. So it actually, there is opportunity to use cattle grazing as a way to manage this species. The problem also of having a species that's highly palatable, highly digestible, it can create some concern for ranchers, because Indian grass, as they call it, is highly well liked by ranchers. So when you talk about controlling such species, it becomes more of an issue. So we had like 3 goals as part of this study. The first goal was to assess the cost effectiveness of cattle. I mean, that's an overarching goal, the cost effectiveness of using cattle grazing and chemical treatment to control this particular species. We had 2 main questions. So others grazing, which are symbolized as G, and herbicide application H, affect West Indian marsh cover and biomass. And we expected that both grazing and herbicide will be efficient at reducing West Indian marsh grass, but that herbicide will be the more efficient, effective way to do it. The second question is like, how does it impact species richness in these wetlands? So we expected that cattle exclusion would reduce species richness, because we have found in the past that fenced wetland that had been invaded by West Indian marsh grass, now you have just a West Indian marsh grass wetland with very low diversity. And we thought herbicide will increase species richness, and we didn't expect really much of a difference between the grazing and the herbicide treatment on the species richness. And the last objective that I'm not going to talk about is to look at really the cost effectiveness, so the cost of applying those different treatments. And that's the work we're doing with USDA NRCS economist Jessica Bertin that we're going to focus this year on. What about our sites? So we decided to select multiple sites and not just work at Buck Island Ranch. So we set up multiple plots, 4 plots at Buck Island Ranch, 3 plots at the station. You might have seen them at the reserve in Francis Creek and Mary's Creek. And we also selected a site that wasn't part of Archbold, and we thank Carrie Lightsey for that. So we have set up 3 plots at Santa Rosa Ranch near Elwyn Park. The criteria for the selection was that it had to be cattle ranches. Ideally, we wanted sites to be embedded in wetland restoration easement. So it's the case at the reserve, Santa Rosa, and 1 site at Buck Island Ranch, but all 3 sites at Buck Island Ranch are not within easement. And of course, we wanted to focus on seasonal wetland, but Santa Rosa Ranch is more like a floodplain system than a seasonal wetland, seasonal depressional wetland. And what we did at each of these sites, we set up multiple plots organized this way, where we have, it's a 10 by 10 meter plot. We separate it in 4 sections. Each section is applied a specific treatment. So we have graze, ungraze, graze plus herbicide, or just herbicide. We started the project in 2001 by setting up those plots. We had a first application of herbicide on the first year. We did some short-term survey following the herbicide application and long-term survey. And in the second year, we applied another set of herbicide application if needed. As far as our measurements, we did some, of course, vegetation surveys using like meter square plot, as well as recording all the species found in those plots. We did that we had baseline survey, a survey after applying 80 to 120 days after chemical application and more than 200 days. We repeated that in 2022 as well, but I'm not going to show you those here. And for the species survey, we just use like classic 1 meter square plot. We record species presence and cover. And we also built some allometric relationship between vegetation height and biomass, because our plots are so small. We didn't want to use like destructive method to quantify biomass. So we built those allometric relationship, and they actually work pretty well because we can explain 80% of the variation in biomass by just measuring plant height. And over the course of the experiment, we took photo points to have a visual way of quantifying this. So, and the first result I'm going to talk about is I just want you to know that Western marsh grass is highly productive. This is a picture showing you just after we set up the plots at the start of the year, and this is like maybe 4 months. So this is about 1 to 2 tons per acre that is produced by these species. As far as our results over the short term, as you can see here, this is what's the difference between the graze and the fence plot. So as you can see, grazing allows for managing the cover of that species. And I separated here the bar plot, so it's the Western marsh grass percent cover at each of the 3 different sites, because we wanted to separate each site because they show different things. So Buck Island Ranch is where we observe very efficient effect of cattle grazing, and Santa Rosa Ranch, not as much. As you can see, this bar here, the fence bar plot here compared to the graze here are not significantly different. The herbicide treatment was particularly effective at Santa Rosa, where we didn't observe any West Indian marsh grass after herbicide application. And we think it's probably due to different type of soil, which are more organic, that might allow for better transport of the herbicide. Over the long term, the story is a bit different. We still observe that if you fence, you're gonna increase West Indian marsh grass cover. In fact, we have an 80% increase in cover and biomass in those plots after you fence it after a full year. The lowest West Indian marsh grass cover was observed in the herbicide plot, and as you can see, even a full year after treatment application for Santa Rosa, both graze plus herbicide and fence plus herbicide had no amount of West Indian marsh grass, so it was particularly effective there. However, we still observe some resprouts at the reserve and at Buck Island Ranch, so which suggests that more treatment application will be needed, and so 1 time or even 2 time application might not be enough. Yeah. So the conclusion after 1 year of treatment. So herbicide and grazing both reduce West Indian marsh grass cover on the short term and after a year. So as I mentioned, fencing, for example, if you fence, if you apply nothing, you will see a decrease of 37% in species richness and an increase in 80% in biomass of Western marsh grass. So if there is something I would like you to keep from this talk is that if you do absolutely nothing, you're gonna end up with a big problem in those wetland restoration easements. So you have to do something. Herbicide was the most effective. It actually reduced Western marsh grass by 80% in our overall across all plots that we use. Grazing does reduce it as well by 48%, but as you can see, herbicide application is the most effective treatment. In many sites, we observe resprout, suggesting that you will have to go back to the same plot and apply herbicide again. And the one thing about grazing is that you will also observe resprout there because Western marsh grass tolerate grazing. And so you have to keep grazing on a regular basis in those sites. So you might get be okay like herbiciding like 2 or 3 times, but grazing will have to be continued, like rotational grazing continuously in these easements. And that's about it. Yeah. So and I have a little bit of time. So earlier I mentioned that we've not much. So I just would like to thank you, Akeem, Alan, and all the people that are building those plots. And I was just mentioning that Akeem, through his research internship, actually discovered or found out that this species was actually more of a transient seed bank than a persistent seed bank, because we didn't observe it really much during seed germination experiment from soil from these invaded wetlands. Thank you.
Thank you.
One quick question while we're changing over the slides. Reed, you had your hand up.
Gregory, is there any evidence that cattle could also disperse the grazing of it?
So like it's through like they can disperse it. I don't think it's been documented like scientifically, but that's kind of a common assumption that they would disperse it through just like breaking it into like. There's absolutely no evidence that the seeds, if they are eaten, will remain intact after being digested or like and germinate from the cow hands.
Okay, so we're very close to being on time, so we're going to press ahead. Thank you so much. And hand over to Hannah. I'll let you introduce yourself and give your title. So thanks.
Thank you. My name is Hannah Bowen. I'm the newest RA in the plant lab. And today I'm going to tell you about, I think, the only project I've been in charge of so far since I got here in October. It's about fire management and the success of introduced Chrysopsis floridana, with the title of long-term success of Chrysopsis floridana introductions depends on continued fire management. It's a collaboration between myself, Cheryl Peterson, who I hope is in the audience and will be available to answer questions about this afterwards, Eric Menges, and Aaron David, who is also here. So just to begin, a little species profile of Chrysopsis floridana. It's a short-lived perennial. It's in the aster family or the composite family. So like the center of the flower is actually lots of tiny little flowers. And it occurs in shrub or shrub-like areas. And it is federally and state endangered with the possibility of a state delisting. Its range only occurs in 5 counties in southwest central Florida. As far as we know, the true range isn't truly known. And it was federally listed in May 1986. And like I said, it's up for possible delisting. And there's currently only about 30 populations. So like I said, this research and conservation efforts has been a collaboration between Bok Tower Gardens and Archbold Biological Station. And Bok Tower Gardens has been with this project since 1986, and then Archbold has been working with it for about a decade. And primarily, Archbold has been involved in the demographic evaluation. And specifically in this talk, I will touch on population health and comparing introduction strategies. So the sites, the way that the sites were selected for Chrysopsis floridana introductions was looking at the historical range of the plant and also looking at suitable and managed habitats. I just want to emphasize the management part. And here, the sites that Archbold has worked with and that's presented in this talk are bolded. And you can see in the graph on the left corner, the bars are color coded by site, and then the y-axis is just the raw numbers, raw population numbers. And basically the point of the graph is to show that since the introduction efforts started in 2008 up to 2015, we did see increases. And in 2016, Archbold and Walk Tower Garden changed and streamlined the protocols, the demographic monitoring protocols, specifically natural and introduced populations were compared using demographic analyses, resulting in this paper that came out of those efforts. And then after 2019, the monitoring was continued, again, to track with the goal of tracking response to management. So that brings me to my research questions. The general question is, how does fire management affect the long-term success of Chrysopsis floridana introductions? And then the specific question is there a significant relationship between time since burn or time since fire and the measurable demographic variables that would indicate long-term success of these introductions? So the methods that were used are typical demography and stage class data collection. I won't go too much into it, but in the plant lab, we call full demography level 3 and stage class level 2. So using flags and tags for demography, we take different demographic variables. And then for the stage class or level 2, it's just raw counts. So those don't have tags continuing over time. And using demography and stage class data from since 2017, so for 5 years, we're able to calculate these demographic variables, reproduction. So number of flowering heads per meter squared per year can give us an indication of reproduction. Number of new seedlings per meter squared per year is a proxy for recruitment. And then we can also track population size by looking at the percent change of plants over time. And so I wanted to see the relationship between those demographic variables and the time since last fire. So with the help of Aaron David, we looked at some linear mixed models using random effects to control for the different sites. And the result was that time since burn decreases recruitment. So on the x-axis is the time since burn in years, and then on the y-axis is the number of seedlings per meter squared per year. The colors indicate the introduction site, and then the shapes indicate the year. Overall, you can see visually that there's a pretty clear downward trend for the number of seedlings and the time since fire. And this model did give show that there was a significant effect. The R squared is pretty low, but for biology, I'm told that that is acceptable. And then so looking at another demographic variable, which indicates reproduction, number of flowering heads per meter squared per year and time since burn, we see a similar downward trend. There's of course a couple outliers. And we see an even more significant relationship between time since burn and number of flowering heads. So this graph, I want to take a little bit of time to explain it because there's a lot going on, but the general takeaway is that managed sites or sites that receive burn are generally favor growth of these plants that are introduced. And so The color coding again is to indicate the site, and then the line type indicates whether that site was burned at all between 2017 and 2022. And so, and then the point, whether it's filled in or not, will indicate when the burn actually occurred. So if you just first look at all of the solid lined sites, you can see that they're the ones that have the most, oh, I forgot to tell you what the y-axis was. Okay, let me start over. So the y-axis is the percent change in relative density in number of plants per meter squared as compared to 2017 when we started monitoring. So what this is showing is how does the number of plants per meter squared change? How has it changed since we started monitoring, since 2017? So that's why all of the lines begin at 0. Okay, and then now if you compare the dotted lines and the solid lines, you can see that overall the solid lines show generally more number of plants, and then also after a burn, which is the solid point, there's a general trend to increase in the number of plants per meter squared. Of course, there's some exceptions. In 2019, it looks like pretty much all of the sites increase in number of plants. So that could have to do with a lot of different factors, maybe weather or some other general trend that affected all of the sites. And then just another way to look at that data, but only for 2022. This is the ending percent change in relative density since 2017 for this year. And the bars that are outlined in red were burned at some point. And so you can see that the ones that were burned, with the exception of Portal West, have all increased positively since 2017. And the ones that weren't burned, Perico, Wheaton Island, and Cordell East have not increased. Well, Cordell East has a little bit, but not in a big way. So in conclusion, controlled burns favor the long-term success of introduced Chrysopsis floridana populations. And another way to say that would be, or bringing it back to introductions, introductions are only as successful as the management that follows. So my favorite slide is the acknowledgements. There's so many people involved in this project. And like I said, it's a running project since 1986. So of course I'm gonna miss some people. But I will first shout out to Cheryl Peterson and Chico Rivera from Bok Tower Gardens, who I had the pleasure of working with this year in the field. Of course, Dr. Andee, the plant ecology program director, who guided me a lot through the statistical analysis and research question. The previous plant lab director, Dr. Eric Menges, Dr. Scott Ward, who did a lot on the paper that I mentioned. The current plant lab, Dr. Sterling Heron, Amy Naccarato, Iris Kennedy, Antonia Jordan-Millay, Jeffrey Dazal, and Layla Wilson. Hey. And then Liz Childress from Pinellas County Parks and Rec, who's very kind to us when we visited Wheaton Island. So that's the end, and I hope there's some time for questions. Cheryl Peterson, I hope, is here. Are you there, Cheryl? She knows a lot more about the project, so I want her to be available. Yeah.
Maybe Laura can bring that up somehow. So we do have time for questions. Thanks for a great talk. And we'll start with Deborah Mitchell.
Hi, I'm really curious to hear how the pandemic affected the monitoring of the sites, if at all, in your case.
Yeah, that's a great question. I have no idea. So maybe Cheryl could answer, or Erin, do you know?
The data were collected, so I guess not.
I guess I could answer in general. Archbold, at least on our end, continued with all field work. I don't think there were any projects. It was a big struggle and a lot of complications with vehicle transport and working in the field. But if anything, field work was the one area where we were able to persist. I think others would generally agree. So next question. Yes, next.
Hi, I just had a question on your thoughts as to why one of the sites.
Speak loud.
Sorry, for one of the sites, it seems the burning actually had a negative impact. Do you have thoughts as to why that might be?
Yeah, that's a good question. We were wondering about that too. That's like, it's Cordell West, or it was, yeah. Let's see, it had a negative effect. I don't see from this graph that it had a negative effect, because if you see all of the filled in points, there is a general upward trend. But I think maybe you're referring to this one where Cordell West was burned at some point, but then decreased. Sterling, do you know anything about that? I know you've been to, or you know like a little bit about those sites. Sorry to put you on the spot.
I don't know about that particular population, assuming when plants got burned in that case, but I guess.
Yeah, what about you, Iris? You were there recently.
Well, we did find a lot of like burned flags and burned plants. I wonder if just the bird, we just got like unlucky with it hitting more of like the reproductive.
Or like the burn intensity
Yeah.
varied, yeah. Yeah, that's like the disadvantage, I guess, is that we don't have like a lot of detailed information about the burns. We just know like when they were, but we don't have information about the intensity, the patchiness, frequency, things like that. So it's hard to say like what specifically got burned and what didn't.
Thank you.
Yeah, thank you.
So why don't we ask, see if does anyone online have a question, Laura?
Cheryl had the question that she didn't hear the question, which I think was, did the pandemic affect the sampling and the research?
Yeah, Cheryl, the question was, how did the pandemic affect the sampling and field monitoring? And we answered that. Any other questions? Well, if not, thank you very much.